We are starting. All right, so welcome everybody to another StaPH-B call. It's been a couple months since we've had a call. It's been a busy time for me, so I apologize for that, and I'm sure it's been busy for everybody else as well. But we've got a really exciting speaker on today that I'm really excited for, and I think that everybody else is going to really enjoy as well too. So it's kind of a funny story. My mom, of all people, is our speaker's neighbor. And so she was walking around the neighborhood and they've interacted before and she was like, oh, you've got to get this you know, our neighbor on one of your calls. She does this really amazing work and stuff. And I was like, oh, okay, Mom, you know, yeah, that's great. But then she actually sent me Kate's information and we started chatting a little bit, and Kate is actually doing some really amazing stuff, and she works for a company that's developing a vaccine. And so I think that it's going to be a really interesting call today for us, because on the public health side, we often get to work on a specific kind of area of public health, normally more kind of surveillance and the testing kind of areas. But I think it'll be really interesting for us all as a community to see what's going on the kind of vaccine development side, and also kind of to see some of the maybe different ways that the bioinformatics is used in different areas that are also impacting public health. So without further ado, I will introduce our speaker, Kate Broderick. So welcome, Kate. Sorry, I don't have a introduction information or anything like that. So if you want to introduce yourself and your team, you can go ahead and take it away.
Thank you so much, Logan, for having myself and Jean and Charles here today. It's such a pleasure. And you're absolutely right, Logan. I thought, and I hope that over the next however long we chat for, you know, you guys are doing such vital public health work. And sometimes I feel like we don't always interconnect well enough, even though what we do is critical to what you do, and what you do is critical to what we do. So hopefully this kind of can build some of those bridges, and we're definitely completely open for questions or discussions, you know, as we go through this. But, you know, from hearing, Logan, what you and in your lab do every day, you know, you're literally on the spear tip of what's going on for COVID at the moment. We're kind of also completely overtaken by COVID at the moment, but on the vaccine development side. So hopefully the discussion today will kind of see where we kind of both interface and where there's synergy really between what we both do. So I'm going to talk kind of on the high level what we do when it comes to vaccine development side, but I've also asked Dr. Charles Reid to join us, who's our sort of bioinformatics guru, and he'll talk through really what about our vaccine development uses those aspects. And then I've also got Dr. Jean Boyer here on the line. And Jean, I don't know if you can stay the whole time. I hope you can, but if you can't, it's fine. Who heads our bioanalytical work here. So, you know, Jean is completely in charge of all the receipt of all our clinical samples from our clinical trials, performing all the assays on those, and really getting readouts on basically how our vaccine development's going. So I thought the three of us could kind of tag team today, and it might be of interest to everybody from all your different walks of life. So let me go through our slides, and please feel free to make this interactive. Please stop me or the rest of us at any time, and we can talk through it. So Charles, Jean and I all work for a company called Inovio. I'm based, well, I have the luck of being based in San Diego. Charles is based in our East Coast side of things. And our speciality is DNA-based medicines or vaccines. Now, everybody and their uncle now, as of a year ago, knows what an RNA vaccine is. And that's wonderful, because you've seen what, you know, the impact of the RNA vaccines have had from an efficacy standpoint, which is absolutely remarkable. We as a DNA vaccine company have a lot of similarity to RNA vaccines, but there's some sort of subtle differences. And I'm kind of going to talk through that from that perspective, but with an eye on kind of how do we apply all these differences and all this technology in an ongoing pandemic setting as we are right now. So This slide is a little bit corporate, and I apologize for this. I just kind of pulled some slides from different decks, but I wanted to give you kind of a a 40,000 foot view of what we do basically at the company. So because we don't just make vaccines, we actually do DNA encoded monoclonal antibodies as well. So we call it DNA medicine. So we have 15 ongoing clinical programs at the moment. As you can imagine, for the last year, we've pretty much, most of our bandwidth has been sucked up by COVID. But, prior to January 2020, we had a really robust pipeline, and I'll talk a little bit about that too, just to kind of set the scene that it's not just a COVID company. So we use DNA plasmids, which are circular pieces of DNA, and we deliver them into patients' bodies using delivery technology. So Charles is going to talk about how we actually design those plasmid pieces of DNA, and we'll talk kind of about our experience with COVID, because obviously in January 2020, when we started this, we had one sequence, and that was the Wuhan strain. Today, that's completely different, and as such, we've utilized all the GISAID website information, and we've actually been able to come up with a more pan COVID-19 approach. So, early on, we started with a single match strain vaccine. Now, of course, we have the benefit of a huge dearth of information about different viral variants. And as such, this can kind of be an evolutionary vaccine development, in that we can add all that information in to ensure that we have better vaccines moving forward. So we're talking about a prophylactic vaccine when we're talking about COVID, but we also can utilize our technology for therapeutic vaccines, and we're treating all sorts of different types of cancers. We don't have an approved product per se at the moment, but we've treated over 3,000 subjects and patients in clinical trials. And I'm going to talk through some of the other benefits of our technology as we move through the slides. So this is kind of a little bit of these are some areas that our vaccines work particularly well in the green shady area. And then on the right hand side, it's how do you then, how does that play in a pandemic situation that we're in at the moment? And, I'm pretty 100% sure I don't need to tell anyone on the call moment that this kind of sense maybe at the moment, that because so many of us have been vaccinated, the end is in sight and the end of the pandemic is in sight. I can't see how that gives me huge levels of anxiety, because I don't concur with that mindset at all. And we only need to look to the situation in India to see how, the variants can completely turn something on its head. So I really am concerned about this sort of sense of, oh, we're vaccinated, we're kind of good to go. I don't subscribe to that at all. So in the green, DNA vaccines in general drive CD8 responses. And I'm assuming some of you guys are kind of familiar with the immunology here. The standard kind of immune response that you're looking for a viral threat would be an antibody response. So that's how you hear about neutralizing antibodies, all the rest of it. However, antibodies in general are really susceptible to mutational changes, because that's the whole mechanism of action of an antibody. It's very specific. So if you start changing some of those base pairs, which obviously modifies the protein sequence, then that's going to impact the efficacy of a vaccine. And so that's why we're seeing some issues with the variants right now. So one aspect of our vaccine technology is that it not only drives antibody responses, but also T cell responses, which are much less impacted by subtle sort of mutational changes. We also, I think, all need to embrace that a one and done vaccination regime is not going to cut it for COVID. I think, we all probably are all accepting now that this is going to be, at least require multiple boosts, if not annual boosts, potentially, ad nauseum, and it should be thought like flu. So as such, we need to think about vaccines that can be re-administered every 6 months, every year, forever how long is required. And certain vaccines are very suitable for that, and certain vaccines are less so. For instance, the viral vector vaccines, the AstraZeneca and the J&J, because they are made of viruses, the body does what the body should do and kind of raise immune responses to those vector vaccines themselves. And so over time, your ability to respond to those becomes less and less. And for those of you, and I'm certainly one of them, who received your mRNA vaccine, you might notice that between the first and the second immunization, the side effects got slightly worse. Certainly, I didn't have a huge amount of side effects with the first one, a bit of a sore arm. Second one, I had much more sort of systemic fevers, that kind of thing. The potential is that could continue to increase over time. So as you get your third, your fourth, your fifth, that continues to get worse, which would really kind of limit then the compliance and the uptake of vaccines in a reboosting. So we need to just think through what vaccine technologies are appropriate for the initial primary response. And clearly the EUA vaccines have done an amazing job there, but they wouldn't necessarily be the right ones for then seasonal boosting. So we'll talk a little bit about safety and tolerability. And then one other thing I did want to talk about, and it really came up quite early on in the discussion of implementing vaccines, was the incredible complexity that transportation, storage, logistics takes on getting vaccines out to everybody. And we're talking about a US setting here. And we have the most resources on the planet, right? And even that was difficult, getting -80 freezers into every doctor's office. And you guys probably know more than anyone else how big a bloody -80 freezer is. So getting that into, a small rural doctor's office is going to be really complex. So you imagine trying to do that in sub-Saharan Africa. I mean, it's frankly impossible. Let's be honest. So there are a lot of limitations that are put in place basically due to the storage conditions of the vaccine. So for us, the DNA is an incredibly stable molecule. You could essentially sit with vaccines on your desk in your office for a year with no impact on stability. So there are so many nuances to think about when we are thinking about a global vaccination campaign, as we absolutely have to when it comes to COVID. So I'm going to move on. So I've talked a little bit about our technology, what we call our platform. So, we're talking primarily today about COVID, but frankly, prior to COVID, we were working on Ebola, we were working on MERS, we were working on SARS, on Lassa, on a variety of different infectious diseases. And as such, and again, I don't want to be a negative Nancy, but when the next pandemic hits, not if the next pandemic hits, when the next pandemic hits, we need to have technologies that can be ready to pull the trigger right there, right then, be able to ramp up really quickly from a design perspective all the way up through manufacturing and implementation, so that we are not kind of always playing catch up. And so that's really where we feel we bring a lot to the table, because it's so fast, and Charles is going to talk this through. But in the case of COVID, we received an email from the Chinese authorities on January 10th, I think it was 2020, with the genetic sequence of the Wuhan strain. Within a matter of hours, we designed our vaccine, immediately went into GMP manufacture, and in parallel, we were running all the appropriate preclinical safety animal studies, and then it was in the clinic within 83 days. Now that speed is unprecedented, and I know that's a way overused word at the moment, but for us that was unprecedented. And I actually think we could do faster than that now that we've learned from that experience. But when we're talking about pandemic outbreaks, it's that really that speed that we're going to need to be able to implement on a regular basis to allow us to be able to kind of to really meet these threats full on. So with that, I'm going to pass over to Charles, who leads our antigen design process, to talk you through kind of basically how do we come up with the designs of these vaccines.
Thank you, Kate. First off, I do want to say I appreciate the time, and I appreciate everybody being here for this. If anybody has questions, please do feel free to interrupt me, or if you'd rather wait till the end, that's your call. So as Kate mentioned, I'm going to go over at a very high level our design process, and I'll specifically deal with some of the issues we had with SARS-CoV-2, but realize that what I'm talking about here does have very general applications to many different possible scenarios, as Kate previously pointed out. The antigen design team actually supports the whole R&D group with specific DNA-based designs that encode for either antigens, our DMAb in vivo antibody expression platform, potential adjuvants, or other types of proteins and reagents for experimental purposes. While the team does have a great deal of experience in antibody design and engineering, I thought I would take a few seconds here and actually highlight our vaccine design platform, the SynCon platform. In terms of our approach with vaccine designs, we attempt to design a molecule that is going to have a broader representation of the possible antigens that could be seen in a patient that may have different strains being present in either regional or global circulation, where we may not be sure exactly what strain of a virus we might be encountering. So the SynCon process attempts to capture the broader applicability of certain of these antigenic epitopes. We take samplings of sequences that are available to us and generate a consensus sequence. Now, as you can imagine, it's entirely possible to take a large sampling of sequences, generate a consensus, and at the end of that process, have something that is not necessarily representative of any of the molecules that you are looking at, yet, however, contains certain conserved regions that, while not completely mirroring any one strain or any one sequence, actually overlaps with multiple of them. The goal of the SynCon process is to develop that type of broad-based antigen that would potentially have a longer therapeutic window of efficacy instead of an approach that would be a specific strain matching event. As strains would differ, rise or fall, that specific strain matched vaccine might become less relevant. With a SynCon design, we're attempting to not just apply the general approach of a consensus sequence, but also applying knowledge that we bring to the table in terms of biofunction of the molecule, in terms of known B or T cell epitopes, and in terms of our own analytics on these sides, to try to capture what would be a broad representation of antigens, but is not necessarily a matched sample for any one that we're looking at. Obviously, once we have our sequences designed, we have to encode them into a DNA format. Inovio actually has a proprietary gene optimization algorithm that we've developed in-house, and that development was actually driven by empirical data that we've generated in multiple in vivo models. We are able to take our DNA sequences, rapidly convert them using this GOAL algorithm. And once we have these finalized sequences developed, the last part of the process would involve the insertion into the plasmid and the plasmid design itself. As Inovio is a DNA-based therapeutics company, with the plasmid DNA being half of the core technology, the delivery methods of electroporation being the other half, the plasmid design is actually a central part of that process. And we take our transgene and actually insert into one of a variety of different backbones that we use, with either single promoters, dual or bidirectional promoters. And we even have design specifications to allow us for multi-cistronic expression. So a single promoter could actually drive the expression of more than one antigen. In terms of that design process, promoter strength, as well as specifics in order and the type of fusion elements that are used, we can also control expression levels in terms of plasmid design. And that completes the design process that the antigen design team would do. Kate, if you could advance to the next slide, please. What I wanted to do before I talk about what happens after that was actually take a second and highlight that gene optimization algorithm that we use. Again, this was an algorithm that we had developed in-house, and it was driven by data that we generated ourselves. As you can imagine, when people talk about gene optimization, it means different things when it's said by different individuals. Companies that are involved in DNA synthesis often talk about optimization, and what they usually mean is a sequence that's been formatted ideally to be synthesized in whatever platform they happen to be espousing. When we talk about gene optimization, we're talking about optimized for Inovio's needs and uses. And this is specifically for immunogenicity, expression, and biofunction. We engaged with the R&D team to actually experiment with different parameters of codon usage, of RNA design. And from those experiments, we're able to choose the most relevant parameters that we then put into a computer algorithm to allow us to make specific codon selections optimized for our need at hand. One additional feature that we have with this is that it's not an algorithm that's written in stone, but we actually have parameters that we can tune and change for specific needs. So the goal algorithm is a tool that we use now. It has great utility, and it also has the ability to grow and change as specific applications come up in the future. If you could advance to the next slide, Kate. I kind of left things hanging when I talked about the end of plasmid design, but I want to mention also that the antigen design team works very, very closely with the plasmid production group. And staying vigilant on the next steps of the process actually helps us to improve our design efforts to make sure that what we are indeed designing is of the best possible quality and is also sensitive to downstream needs for scale up, for production, and for purification. We actually have people on both antigen design and plasmid production to provide that level of insight that is really only possible when you're existing within one of those spheres or the other. We have people that can straddle both of those worlds. And the production of the DNA is really the first step in our screening and testing processes. So the antigen design team really seeks to provide rapid solutions to design questions. We apply our computer algorithms and our design processes, as Kate mentioned, very rapidly, within hours. And we're able to quickly drive to generating that plasmid DNA design for synthesis and subsequent downstream testing. Thank you, Kate.
Thanks so much, Charles. That was great. So I'm going to zip through the rest of these slides fairly quickly, and we can certainly kind of return to any bit. I thought something that might be of interest because it's kind of novel to our platform and not something that everyone's super familiar with is our delivery technology. So because we use DNA and DNA is a pretty big macromolecule, you can inject it into the skin or into the muscle, but generally the efficiency of the uptake into cells it's pretty low. So we use an enhancing technology to basically push it through the plasma membrane, make that a little bit more efficient. And you're looking at this device here, and just to give you a sense of scale, it's about the size of an electric toothbrush. We now deliver all of our infectious disease vaccines to the skin, so we don't do intramuscular anymore. So you get about 0.1 mils, about 100 microliters of vaccine delivered to your skin. Then you hold this device basically up against the skin. It delivers very small, localized electrical field pulses, which basically cause the plasma membrane to act like a capacitor, and it basically changes the dynamics of the membrane for a very, very short periods of time. The DNA is able to shuttle into the cell. As soon as the electric field is released, then the membrane reforms. So down on the bottom here, I didn't mean to do that. You can see kind of the benefits of without the delivery, or we call it electroporation, you really get very little uptake of DNA. With it, you can see it really, the cells take it up very well. It's very easy to use. We've done extensive usability studies with it, very safe, very tolerable. It's kind of a weird feeling, I guess I would describe it. Doesn't hurt, but it's an odd sensation, almost like kind of somebody tickling your skin. I would describe it as the most. And, you know, but we're very aware that anything new takes some, you know, some mindset changes. You know, needles and syringes have been around for about 300 years. So we kind of think of this as, you know, a 21st century approach to vaccine delivery. So this is just a quick snapshot of what we do when we're not working on COVID-19. My particular passion has always been on the infectious disease side of things. We were working on several other coronaviruses before SARS-CoV-2 hit. Also Lassa fever, again, gets very little press, but it kills way more people in Africa than Ebola ever does. And even though we have some decent Ebola vaccines, you know, they have their limitations too. So we're working on those as well. And then you can see, you know, on the oncology side, on the HBV side, we have a lot of other activities going on there as well. So for COVID specifically, as Charles mentioned, we kind of started in January with one design, and I'll talk a little bit about the new design. But basically, we have completed our phase 1, our phase 2 is ongoing in the US, and we're going to give an interim readout of that relatively soon. And then we're hoping to start our phase 2 globally. And I say globally because really that's where we're seeing the highest levels of infection rates at the moment. Thankfully, you know, the US infection rates are really kind of coming down very nicely at the moment, but that is really absolutely not the case in the majority of the rest of the planet. So we're going to be taking our phase 3 efficacy trials to countries where that is an issue. And we've been really working hard on manufacturing scale up. I mean, you can have the world's most effective vaccine, but if you can't produce relevant numbers of the doses, you know, it's kind of not really worth your effort to move ahead with that. So I've kind of talked through some of the differentiators. We have a really nice safety and tolerability platform. You don't see that kind of increasing reactogenicity as you get more and more doses. And we think that's really important, especially as we're almost certainly going to need booster doses with this. We have really nice immunogenic profile, antibodies and T cells. And we've got that, you know, really preferable thermal stability profiles so that, you know, it's certainly in the case of, just to give you an example, in the case of Ebola in the DRC, those were all -80 vaccines, and they had to be transported on the back of motorcycles in styrofoam boxes that were filled with frozen jet fuel. So you could only actually, because of the complexities of that, you could only get so many vials of the vaccines in. And if any of you have ever spent any time in Africa, you know, just there isn't transportation infrastructure in many places. So just the complexities of that. We could do it for ring vaccinations for a few thousand people. It was doable, very complex, but it was doable. But to do a whole population, just impossible, absolutely impossible. So we really need to think about those factors when we're thinking about a global vaccination rollout. So I don't know how familiar you all are with clinical trials. This is just kind of an outline of our phase 1 design. So a phase 1 clinical trial always only ever answers the question about safety and tolerability. I mean, that's the primary endpoint of the trial. Is it safe in a small number of people? And then you can ask secondary questions about what kind of immune response does your vaccine generate. So it's very, very helpful work, and it's really the setup that it's required to take it to these much larger tens of thousand people trials that you would need for efficacy studies. So an interesting part about our phase 1, which was all US based, we asked the the obvious questions, the safety and immunogenicity. But then as the epidemiology of the disease was changing so rapidly, we also asked the question, can we give these people a third immunization? And we did, and it was really, really comforting, I guess, if I'm being honest, to see that we could boost these people so well with really no additional side effects, because it looks like potentially natural. The duration of protection from a natural infection is somewhere, we're not exactly sure, somewhere in the somewhere between 6 and 12 months. Well, you know, that's not really ideal. And so a vaccine is going to be able to extend that durability of protection profile way, way further than that. So kind of talking in the concept of VOCs, because, and I'm sorry, I should define that, variants of concern. You know, we all, I think we're all awash in the media with the information about that. I mean, you know, we talk a lot inappropriately about the UK strain, the South African strain, the Brazilian strain, and those are the ones that have been getting a lot of traction. But frankly, you know, you just need to look to India. You've got the B1617 variant that has, you know, I'm not sure it's completely responsible for what we're seeing in India, but it certainly hasn't helped. And let's be frank, as you guys all know, that's going to be what we're going to continue seeing, is these new variants popping up. So we asked the very obvious question, what does our vaccine do against the variants? And that was our original Wuhan vaccine. And we were really very happy to find that on a T cell response, the different variants had no impact at all. So all the variants, we were able to generate the same level of T cell responses. That was great. When we were looking for antibody neutralization, we did pretty well against B117. We had a small drop off, about a twofold drop off, but nothing I think that would be significant. But for the South African, the B1371, 1351, sorry, we had a much more significant drop off. And that was really very similar across all of the vaccines that have been developed so far. That one is just so different that the neutralizing antibody responses were really very impacted. But for the P1, the Brazilian variant, again, we didn't see any impact on the neutralizing antibodies. So we felt that that's pretty good for a match strain vaccine that was designed to Wuhan, that it had some coverage, but it wasn't perfect. And that's certainly the case really for all of the EUA vaccines at the moment. They are not perfect. They're a start, but they're not perfect. So that really kind of catalyzed Charles and his team to say, what can we do with our technology now with all this sequence information to be able to kind of come up with a next generation approach? And as Charles talked about, we've come up with INO-4802, which is this kind of mosaic approach in incorporating the known variants. But we also hope that it might have some predictive power about variants that we don't know about at the moment. Because, you know, let's all be honest, that's exactly, you know, we're always kind of going to be playing catch up if we wait for sequence information. What we really want to do is be able to have some sort of power to be able to kind of cut off a threat before it even happens. And that's what we hope will be the case for INO-4802. And so Charles's team and Jean's team are kind of creating pseudovirus assays. As soon as we get a new sequence, we're generating those assays, and then we're testing those, screening those essentially against our new vaccine constructs to be able to visualize what the impact of the new variants is going to be. So I think this is my last slide, really just kind of summarizing everything that we've talked about. Our technology is kind of considered novel, new, next generation. But, you know, frankly, it's those new next generation approaches that are going to take us to a place where we can be better in a pandemic setting. There's a lot of work to do and we've got a lot to learn. But, you know, I think if you can kind of look at the silver lining of the COVID experience, we've all learned an awful lot. And I think we've all learned what needs to be changed or enhanced or better for the next time this happens. And really, I'll just leave it there and thank you all very much for your time, and Charles and I will be more than happy to take any questions. I think Jean had to drop off, but yeah, if anyone has any comments or questions, we'd be happy to answer.
Actually, Kate, there is one question already in the chat that I was going to field, if you're okay with that.
Please do.
Okay, let me read the question just so everybody's up to speed with the list. It's, how do you deal with epigenetic modifications of target regions or proteins when you design a vaccine? Typically, when we are looking at an antigen that's a potential target for our vaccine design, we do try to understand the biology of the protein that we're dealing with, because at the end of the day, our design parameters are looking at amino acid sequences for then the synthetic encoding using GOL. In cases where the modifications to the DNA could be giving alternate readouts, could be giving different transcripts, could be turning specific genes on and off. Again, at the end of the day, what we're interested in is what is the actual target antigens that we are focusing on. So we do try to be sensitive to those end products. And there may be cases where you have a heterologous set of products being produced, where there is one particular form, other isoforms that could be produced concurrently. And in cases like that, we actually have a few different ways to attack this. One is to design a panel of different antigens based on these different amino acid sequences that have been encoded into plasmids, and conduct in vivo screens to look for efficacy. We also have cases where more than one antigen form could be delivered. We could have a single plasmid expressing multiple different forms of an antigen based on the research, the biology, what is known about the expression of these different forms. We could also generate mixes of plasmids, each one expressing an individual molecule. And in cases like that, they could be screened individually and then mixed based on efficacy results. So from a design point of view, we do try to be sensitive to the actual final changes that are being observed in the antigen that we are targeting. We try to understand the biology of these things, both from the expression of different isoforms, from different genetic modifications that could modify levels or expression. And we try to, whenever possible, develop a broad approach so we have multiple shots on a goal and then a rapid down screen to the most effective single antigen or combination of antigens. I hope that answers the question well enough that you're satisfied with that.
I think that was a fantastic answer, yes. And it looks like that did answer the question. So I am always very fascinated by these talks, and this was a fantastic talk. You guys did an amazing job. So I've got lots of questions, and I'll ask one and then I'll hold back so I can give other people a chance to ask questions too, because I'm sure that everybody has things that they want to ask you about. But so one question that I had is that with the kind of consensus approach that you guys are taking, where you're trying to come up with I guess these epitopes that might not actually exist in real life, but would potentially be something that would elicit responses to different variants and different things. Do you ever find that when you create those particular, I guess kind of like amalgam Frankenstein genes, that they don't actually elicit the response that you wanted at all, and it's like completely the opposite direction?
That was actually a point I wanted to make a little bit earlier. I just didn't phrase everything to get me there. So thank you for this question.
Charles, can I just, and I got to apologize. I just got a text that I need to jump on to another call. Oh. So I'm going to leave you in the very capable hands of Charles to answer all your questions, and say it was a real pleasure to meet you all virtually. And I'm sorry I can't stick on, but my boss is saying, get on this phone call now. So I better do it.
No worries. Thank you so much, Kate, for your time. That was amazing.
Thanks, guys. Take care, everybody. Bye-bye.
So I actually wanted to talk about a lot of different things all at once earlier, but I wanted to stay very sensitive to time because I wanted to make sure that Kate was able to get the whole platform across. But oh, you hit the nail on the head. It's that's actually really what I got to say is what makes the entire SynCon process work. Okay, generating what amounts to a chimeric hybrid molecule is probably my greatest fear. Okay, so we take steps to avoid this. We definitely don't just live or die by what the consensus alignment is that gets spit out of that design process. While that would definitely be an easy thing and make my day a lot smoother, in terms of what we see in efficacy, that is not how we approach this. The SynCon process was actually developed over the course of years, and the research that went into this, and if you look back, there actually is a body of literature going back years on this process. The approach is to generate something that is mostly similar, but has enough diversity that we have a broad approach. So we definitely do not want to, one, disturb or disrupt major known epitopes where there is very little change. Two, we definitely take into account frequency of changes. In other words, if there is a highly unusual change, but is only a very minor representation of the field of variation that's out there. And I'll relate this to the SARS-CoV-2, where Kate mentioned that we've done some work into development of a broadly neutralizing vaccine. We actually looked at this type of information. We looked at not only what mutations are happening, where are these variations occurring, but also how often are they occurring? Is this mutation set something that's growing, something that's shrinking in the population? And always taking into mind that in this current situation, another month can go by and all that data can become obsolete. There is inevitable risk in this approach. This is actually, though, one of the areas where I will say we learned from this pandemic. And I want to give a shout out at this point to a bioinformaticist that I work with, Rich Acalia, who has been instrumental in helping us to sort through the amount of information that we've had to deal with during this pandemic. The last point I want to address on this is when we look at variations and changes, we want to do a few different things. One, we do want to see what the overall landscape looks like in terms of the consensus, but we also want to be able to apply knowledge to this. So imagine a protein sequence, or in my case, I'm a structural biologist by training. So I always like to think about this in terms of molecular models. I always try to work in that kind of a space if I'm able to, where we're able to map different types of information. I'm able to map that sequence diversity, but we're also mapping where major important epitopes are, both B and T cell epitopes. But we're also going to do this. We're going to look at those mutations from the point of view of which of them tend to cluster together. And I think this gets to the heart of the problem. If I were to put a blindfold on and just willy-nilly pick representative mutations from variants that are in front of me, I could very well end up with a Franken molecule that is representative of nothing, okay, that has echoes to everything, but is completely ineffective. Obviously, we want to have that echo present, but we want to have that efficacy at the end of the day. And that's where looking at frequency of clusters or groups of mutations that will tend to co-occur, ends up becoming important. And as I mentioned, whenever possible, we do try to look at more than one design in a group and screen and down select. If we're able to design multiple versions of a molecule with slightly different features, if we have an idea ahead of time of, again, to use the SARS-CoV-2 scenario currently inundating us, if we have a sense of variants of concern that may be very, very relevant for us to go for, we then have what amounts to some sort of weighting scheme to apply to the different changes, where we make the changes, how well represented we want particular variants to be within that process. So while there definitely is a purely algorithmic aspect to what we do, there is also rational decision making. And I think in biology, we're all used to knowing that we don't know everything, and forging ahead regardless. The ability to make multiple design variants and to make them quickly, and then to have the team rapidly screen with assays that are useful, assays that have relevance to end stage readouts, and then rapidly down select is really our biggest strength. I can always out-design the ability of any group or team to assay. My goal is not to break the assay team. My goal is to be sensitive to what are the maximum number of variants we can effectively put through the pipeline with the types of assays we're doing, and then capitalize on that process to design a broad array of approaches and then find the one or ones that may work. And in terms of SynCon, being sensitive to co-occurring mutations, being sensitive to changes that might be truly unique to one variant, but completely not present in the rest of the population. These are all things that we will take into account. The single best example within that SARS-CoV-2 spike glycoprotein is probably the RBD domain, that receptor binding domain. And very early on in this process, I made the point to the team that it appeared to me that we were dealing with RBDs that were either of one type or another type, but you very rarely actually saw overlaps. Now I got to say, the landscape is changing and you're starting to see overlaps, some of which are quite unusual. And while that is potentially very challenging from a design viewpoint, I could almost imagine developing variants where you look at RBD changes that could perhaps intercalate, that could perhaps work together to generate a hybrid RBD that might have efficacy. And I say might because I've worked with enough protein structures and sequences to know that the only thing guaranteed is that a lot of what I do is going to give unexpected results. So that's why I would probably work on a panel and have the team screen. And then from those best results, we'd move forward with a candidate. So while I'd love to say, oh, I just hit a button and it's all done. In real life, a lot of rational thought, a lot of application of multiple types of information beyond simply sequence comes into play, understanding the biology of the molecules, and when possible, understanding the structure of the molecules. And at the end of the day, we try to develop what amounts to an antigen with potentially broader therapeutic windows of efficacy. So we're not constantly doing the single strain matched catch-up process. With time, of course, any design in this space could become obsolete, but we want to try to give that vaccine design as large of a lifetime as we're capable of.
That makes sense. Thank you so much.
Oh, you're very welcome.
There are a couple of other questions.
I'm saying.
So yeah, I'll let you kind of read through those and answer those as you see fit, and then.
Okay, I'm definitely not an expert on the formulations of RNA vaccines. I can speak at a very general level with what I've heard or read. I do think there is a certain amount of reaction that some people are seeing, due not to just the immune response to the final antigen product itself, but perhaps due to lipid formulations in terms of the RNA delivery. There could possibly be re-administration or boost problems with multiple administrations. However, just from a very general viewpoint, if I were sitting under the roof of Moderna, I'm sure I would have very active research into novel formulation approaches, into ways of tackling these types of possible delivery shortcomings. And I'm going to say possible because I think there's still very much an active area of research in determining what, if any, kinds of limitations there might be. So I'm definitely going to couch my language very carefully there because I am not the expert in RNA therapeutic formulations. I believe there's another question. Let me make sure I didn't miss. Okay, yeah. So great talk. I'm curious, with stimulating both humoral and cell-mediated immunity, do you see any potential in eliciting response from NK cells through ADCC to leverage a broader response with antibody targets compared to just neutralizing Abs? That starts to touch on ideas and concepts related to chimeric antigen receptors, engineered cells, possible the T cell engager space. Inovio actually has interest in things besides SARS-CoV-2. We have experience in developing in vivo monoclonal antibody expression. We have experience with in vivo bispecific molecules. Potential of engaging NK cells. That's not something we ourselves have worked on. In a previous life, I was actually involved in work like that, engineering certain immortalized cell lines for an approach similar to chimeric antigen receptors. And you're actually seeing some representation of that today. In terms of an in vivo delivered product that could do that, is everybody here familiar with bispecific T cell engagers, BiTEs, or what else could I call them? Dual specific single chain variable fragments. Is that type of molecule familiar to everyone? Maybe I should just explain it very quickly just to, so there is a company, Macrogenics, and they were pioneers in the developing of a linked pair of single chain variable fragments, SCFVs, that were targeting two different cell surface receptors. The concept is that by administering this small protein molecule, this T cell engager, they're able to create a bridging molecule to actually link T cell to a tumor. The concept is very similar to chimeric antigen receptors, except instead of taking out T cells from an individual, transducing those T cells with some sort of tumor engaging receptor and putting them back into the patient, the idea was to use the body's own endogenous T cells already present, no leukapheresis, no transduction and logistics of transport, but introduce a small protein that would actually be a bridging molecule to do this. That is a bispecific T cell engager, or BiTE. There are other versions of these molecules. Other companies have come up with a similar molecule that has some disulfide bonding in it called a DART. And there are actually quite a few other of these formats. You'll see even trikes, and everybody has their own buzzword for these types of formulations. But the concept of being able to bridge a target cell to an effector cell with a small protein that can be administered is really there. That could be something that could be administered with, let's say, an in vivo electroporation platform. I will say that the recombinant versions of these molecules, because they are single chain variable fragments, they are the FV portions of antibodies, and they're made up of heavy and light variable domains with a linker in between them. The molecules are, by their nature, somewhat unstable. They will tend to fall apart and aggregate over time. And administration of them is typically done with some type of infusion pump over several hours or even a day or two for individuals. And as you can imagine, that definitely complicates delivery. An in vivo expression platform of that type of molecule could potentially overcome some of these logistical delivery issues. And I will say this from previous work that we've done with in vivo expression of antibodies, the types of challenges and advantages and disadvantages with recombinant antibodies that are made in some sort of suspension cell line, purified, concentrated, stored in concentrated form, or lyophilized. The types of design considerations, the types of amino acid changes that have to be made to the antibody, a lot of those rules actually don't apply with in vivo expressed antibodies. A lot of my experience with recombinant monoclonal antibody design and development, I actually found myself in some cases relearning what is most important when we got to the point of in vivo expression of these molecules, because we're not dealing with purification, concentration, long-term storage, harsh buffer conditions that might occur during that process. We're not dealing with lyophilization and reconstitution. A lot of the design considerations, the developability of some of these molecules that I would work on, where I'd be looking for hydrophobic patches, I'd be looking for possible residues, deamidation potential, oxidation potential of these molecules in long-term storage. These are things I didn't have to worry about as much, but I had other things to think about. So the idea of building these types of molecules for in vivo expression, potentially engaging other types of cells, obviously T cell engagers is one thing, but as I mentioned, I'm seeing, I'm trying to think. There is already a name for an NK cell engager, a nice, cute little name. It's slipping my mind right now, but these things do exist, at least at the research level. So
before we go on, I just want to, sorry, just interject really quickly, because we are at time, and so I want to be cognizant of people's time. If you are still available to stay on for a little bit more, Charles, I would love to kind of pick your brain a little bit more, and I'm sure that some people would love to stay on a little bit too.
Bear with me and let me just check an email real quick,
okay.
Because we definitely have okay. Oh, wow. Okay. I am unfortunately going to, what we can do is this, stay in touch. Let's engage another time, because I think there's a lot of really good questions, and I think questions can spur the creative process. I spend my whole day trying to come up with something new, something different, and thinking about questions. It's just some of the best ways I can think of to do this kind of thing. But unfortunately, my emails are piling up. I profusely apologize for that. I truly do.
We were only scheduled for an hour anyway, so I want to thank you and Kate for both being on and speaking. It was a fantastic talk and a really great opportunity for us to kind of engage with a different set of kind of public health practitioners in a different kind of way. And I think that I would love to stay in touch, and I'll send your contact information out to the group, so that anybody who has questions can kind of interact with you and engage, because I think that the conversation could go forward in a lot of ways.
Hey, so, happy to do so, please. Please, forward to whoever has an interest.
All right. Well, thank you so much again. I'm going to stop the recording at least.